Please explain in detail the nervous system. Such as the functional unit is the neuron, resting potential, action potential, impulse propagation, the synapse, and 5 stages of synaptic transmission. The nervous system is a complex network of neurons and supporting cells responsible for transmitting signals throughout the body. It plays a crucial role in coordinating voluntary and involuntary actions, as well as processing sensory information from the environment. The nervous system can be divided into 2 main parts. The central nervous system, CNS, consisting of the brain and spinal cord, and the peripheral nervous system, PNS, comprising nerves and ganglia outside the CNS. The functional unit of the nervous system is the neuron, a specialized cell designed for transmitting electrical and chemical signals. Neurons have 3 main components: the cell body, soma, dendrites, and an axon. The soma houses the nucleus and other organelles, while dendrites receive incoming signals from other neurons. The axon, a long projection, transmits signals away from the cell body to other neurons or target cells. 1. Resting potential. When a neuron is not transmitting signals, it is said to be at resting potential. At this state, the neuron's interior is negatively charged relative to the outside. This is maintained by the sodium-potassium pump, a membrane protein that actively transports sodium ions Na+ out of the cell and potassium ions, K+, into the cell. Additionally, the membrane is more permeable to K+ than Na+, resulting in a net negative charge inside. The resting potential is typically around -70 mV, MV. 2. Action potential. An action potential, or nerve impulse, is a rapid, temporary change in a neuron's membrane potential. When the neuron receives an excitatory stimulus, the membrane's permeability to Na+ increases, causing an influx of Na+ ions. If the membrane potential reaches a threshold, typically around -55 mV, voltage-gated Na+ channels open, causing a rapid depolarization. The membrane potential then overshoots to a positive value, around +40 mV. Subsequently, the Na+ channels close, and voltage-gated K+ channels open, allowing K+ ions to exit the cell, repolarizing the membrane. The action potential is an all-or-nothing event, meaning it either occurs in full or not at all. 3. Impulse propagation. The action potential propagates along the axon from the axon hillock to the axon terminals. This occurs through a process called saltatory conduction in myelinated axons, where the action potential jumps between nodes of Ranvier, the gaps in the myelin sheath. Myelin, a fatty substance produced by glial cells, insulates the axon and increases the speed of impulse conduction. In unmyelinated axons, the action potential propagates more slowly, as it travels continuously along the axon. 4. The synapse. A synapse is a specialized junction between 2 neurons or between a neuron and a target cell. At the synapse, the electrical signal from the presynaptic neuron is converted into a chemical signal, which then triggers an electrical response in the postsynaptic cell. This process involves the release of neurotransmitters, chemical messengers that transmit signals across the synaptic cleft. Stages of synaptic transmission. Neurotransmitter synthesis. Neurons synthesize neurotransmitters, which are stored in vesicles within the axon terminals. 2. Action potential arrival. The action potential arrives at the axon terminal, causing voltage-gated calcium channels to open, allowing calcium ions, Ca2+, to enter the cell. 3. Neurotransmitter release. The influx of Ca2+ triggers the fusion of neurotransmitter containing vesicles with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. 4. Postsynaptic response. Neurotransmitters bind to specific receptors on the postsynaptic membrane, which can either be excitatory or inhibitory. Excitatory receptors generally cause an influx of Na+ ions, increasing the likelihood of an action potential in the postsynaptic neuron. In contrast, inhibitory receptors usually cause an influx of negatively charged ions, such as chloride, Cl, or an efflux of K+ ions, decreasing the likelihood of an action potential. V. Neurotransmitter removal. To prevent continuous stimulation of the postsynaptic cell, neurotransmitters must be removed from the synaptic cleft. This can occur through 3 primary mechanisms. 1. Reuptake by the presynaptic neuron, where neurotransmitters are taken back up and repackaged into vesicles. 2. Enzymatic degradation, where enzymes in the synaptic cleft break down neurotransmitters. And 3. Diffusion, where neurotransmitters simply drift away from the synaptic cleft. The nervous system's complexity is critical for its function, allowing it to rapidly process and transmit information throughout the body. Neurons and their specialized properties enable the generation and propagation of electrical signals, while synapses provide the means for communication between cells. The intricate interplay of electrical and chemical signaling ensures the nervous system's efficient and precise operation.